Good afternoon. We are going to start our second panel, Experimental Science and Medicine, in the framework of transhumanism and posthumanism, reality or fiction. And we will have three experts to talk about the scientific advances and new technology advances to cryogenics and new therapies. We're going to be reflecting with them on possibilities, options, and limits of technologies and perfection or transformation of human features, functionality, or human identity. Right. Today we will have Dr. Nicolas Jouvet de la Barrera, Dr. Miguel Ángel Serra, and Dr. Juan Pablo Romero. We're going to start with Dr. Nicolas Jouvet de la Barrera. He's a doctor in biology, chair in genetics of the University of Alcalá. He has taught at the Madrid University, at the Madrid Poly, at the University of the Basque Country, at the University of Cordoba. Nicolas Jouvet did his postgraduate studies in Cambridge and did a postgraduate course in Colombia. Between 1990 and 1994, he was president of the Spanish Genetic Society. In 1991, the Social Council of the University of Alcalá granted him an award as a researcher, and in 1996, they also granted him an award as a professor. He's a member of the Spanish Bioethics Committee since 2013. He's published over 200 articles, most of them in international journals. Some of his main books are Exploring Genes, From the Big Bang to New Biology, The Fountain of Life, Genes and Bioethics, Stem Cells, New Medicine and the Message of Life. It's a pleasure, Professor, to have you. Thank you very much for joining us. And the title of your talk will be Transhumanism and Enhancement through CRISPR. Thank you. Genetic enhancement through CRISPR. Thank you very much. To Dr. Maite Iglesias, I wanted to ask the technicians to show my slides on the screen, if possible. Thank you. Well, thank you very much, Maite, for this generous introduction. I also want to congratulate the University of Francisco de Vitoria and the organizers of this meeting of the Razón Abierta Institute. In the panel, we have Dr. Serra and Dr. Romero, who are also going to be discussing some very interesting issues to do with the neurology aspects, but I will just focus on the technology of CRISPR, its principles, applications in the human genome, and then some bioethical aspects. In the first question, I'm going to discuss what the CRISPR technology means. First of all, I want to place this technology as the last wave within an activity that has been developed for many decades called genetic engineering. And genetic engineering is something that appeared in the 70s, in the 1970s, and it consists in the manipulation of DNA. It's a direct manipulation of information which is inscribed in the DNA molecules and the sequence of the nucleotide bases of all living beings. We know that the DNA molecule is considered the molecule of life, and it is a molecule that is common to all organisms. What genetic engineering does is try and artificially impact on some sequences of genetic information in order to change those sequences so as to achieve something that is useful. In the specific case of genetic engineering, they started with bacteria, and they came up with genetic bacteria which were capable of synthesizing within their own organism some human drugs, introducing human DNA molecules, for example, of the insulin genes, clotting factors, interferon, etc. Also, genetic improvement was introduced in higher organisms such as plants to achieve high production rates, high quality, resistance to disease, or in animals with, livestock purposes. And then engineering started, focusing on human beings. So genetic editing, or CRISPR, is really a new technology, a new genetic engineering technology, much more perfect, really, and it pursues the enhancement of species and of receptor mechanisms. Basically, it consists in a molecular surgery operation consisting in the elimination or the editing of some region of the DNA involved in something that one might want to modify. So it's modifying the genome with some purposes. So, for example, in a monogenic disease causing some kind of pathology or condition. Now, where does this technology come from, and what does this name mean, this CRISPR name? What does it mean? Well, it is a technology that is inspired in a basic discovery in a natural system, in an adaptive immunity system discovered by a Spanish microbiologist, a chair of the University of Alicante, Francisco Mujica. In some microorganisms, namely the bacteria and the archaea, which are found in some salt beds in the region. And the name CRISPR consists or comes from the description of the area of the genome. It's an area of the genome in these microorganisms which really entails a system of defense against the invasion suffered by bacteria of extraneous DNA. CRISPR comes from clustered, regularly interspaced, short palindromic repeats. So it's a series of repeated sequences. It's those little black boxes which leave colorful spaces between them. And those colorful spaces are the areas called protospaces, and that is where the rest of some DNA of some invading agent appears, and this is introduced inside the bacteria. And when this happens, the bacteria chops up the DNA, and then those little chunks go to those interspaced areas. And then when there is a second or third invasion, the system can react against the attack in a very highly satisfactory way for the bacterium, because it will entail the destruction of the DNA of the invasive agent.
The bacteriophages are viruses that are exactly these agents, the main enemies of the bacteria. After that an infection takes place, those agents, viral agents, what they do is introducing or inject their DNA inside the bacteria cytoplasm. The bacteria reacts, cutting the DNA, introducing those segments inside those spaces that we have mentioned. In front of a second attack, what is going to happen is that the bacteria is going to create some molecules, intermediary molecules of RNA. The RNA is a variant of the DNA with some differences that will recognize by complementation of bases the invasor. The invasor will bind to it, and with the help of a protein that is called Cas, it is going to destroy the invasive RNA. So this is a natural defense system, and this is what Dr. Mojica discovered in those microorganisms. This is the scheme that we are seeing here. In the upper part, we see the virus, how it is being introducing its DNA. And in the image in the left-hand side, we see a second invasion, and how the genome of the bacteria is reacting with those RNA molecules that are synthesized in order for the recognition of the new invasion, and its destruction with the small scissors that we see there. And this is the task of the protein that we call Cas. Well, how did we make the leap to its use in other species and to its use in other species? Well, what happens is that in the biotechnological panorama of current genetics, we know already the genome of many species. And as we have mapped the sequence of nucleotidic bases of the genes in many different species, among them, of course, the human DNA, what we can do is create, as we wish, an RNA that is obeying by base complementation to a zone of the DNA that we want to modify. It is simply using the principles that the bacteria is using, creating artificially an RNA molecule that will be called RNA guide. It's used as a bait in order to go to the genome place where we want to have a bearing on. And adding to that system the Cas protein, we are going to be able to destroy it. To destroy the DNA in the sense of cutting it in order then to repair it. Obviously, for the repair, we need to use a DNA that is given to the cell so that it can use it somehow and can restore the zone that has been altered. So this is a technology that is based on a basic principle of nature and the defense system of the bacteria. The step towards the use in men, and more specifically for the cure or the restoration or the modification, or even better, the editing of some zone of the DNA in the human genome, is due to several researchers, but among them the striking participation of Dr. Manuel Carpentier and Dr. Pertugna, that were Nobel Prizes in chemistry last year, precisely due to the step they made in its implementation for the restoration of a DNA involved in monogenic alterations, especially human monogenic disorders, with clinical targets, of course. So let's see how it is applied for editing this human DNA. I've gone over the technique, but I can't go into details because we don't have too much time. Maybe during the question and answer period, if there is something that is doubtful, we can clarify it. Let's see how this technology is applied to edit the human DNA. It can be done ex vivo or in vivo. We can apply it in the cells of the body itself, or even something better that is normally used. And these are ex vivo system, in vitro system in the lab. What will be done is extract cells from a patient who has a given disease that is due to some alteration in his DNA, because this is what we want to correct. Extract cells from that patient, grow them in the lab, and in those cells that are being cultured in vitro, we are going to introduce the two elements that take part as well in the bacterial system. The guide RNA, an RNA that will be synthesized artificially ex professo to recognize the zone of the DNA that we want to delimit, and the Cas protein or some equivalent one that is going to act insiding on that DNA, cutting it in order then to restore it. But in the restoration, obviously, it will have to be edited, corrected, and put the nucleotidic basis sequence that somehow is going to modify that region and make out of it a region non-involved in the pathology that the patient was suffering. This is what the operation is all about. We have to duplicate those two elements, the RNA guide and the Cas protein, as if they were the hands of a surgeon and the scalpel of a surgeon. The hands to hold the DNA on which we want to inside, and the scalpel that is a Cas protein to cut exactly in the spot that we want to. And obviously we have to know previously the sequence and the merit of the knowledge of the human genome since 2003, as you all know. Once we have made this operation in the cells, as this has been done in vitro in the lab. We verify if the operation is correct. And in the case if it's correct, they are returned to the patient with the aim to restore or somehow to have solved the problem of the pathology that the altered system had. What is the accuracy that the technique has? It's a relatively recent technology because the discovery of the CRISPR system by Professor Mojica took place in 2005. Not too long has elapsed. And the first applications happened a few years later, but we are still starting, so to speak, to use it in applied situations, not only, of course, in the human being, but also in order to modify the genetic information of any organism of whom we know the sequence of DNA that we want to correct, of course. Then, although the precision of this technique is high, it is not. It doesn't go without failure, especially in the recognition of the target of the DNA that we want to be editing, and not either in the repair that has to be done later on to restore or fill the gap that has been left there after the incision that has taken place with a Cas protein. That's why modifications made, uncontrolled modifications that can take place that can alter the genetic information not only of the target area, but also from some other areas. I repeat, the operation is quite precise, but not enough yet subtle in order to be able to use it with complete safety. The probability that mistakes happen, according to some recent papers, varies also depending on the region that we want to modify. There is research from the United Kingdom published in Nature in 2018 that tells about the production of alterations of the type of deletions of DNA and genomic rearrangements that are caused exactly by that technology, CRISPR-Cas, in human cells cultured in vitro. But in 2019, a new technology appeared, a modification of the technology. So it's not too long ago. It's called prime editing, that is pairing one step. It's less complex than the initial techniques, and it allows greater precision. Even so, there is still a certain margin of unsecurity. It's not an absolutely specific, precise technique. And a word of caution, the technology is not the same to implement it in any tissue or any type of cell. In the human case, when it's done on somatic tissue cells maintained ex vivo in the lab, as we've said, there will not be great problems of safety, because if the editing is not satisfactory, the cells are not going to be implanted in the patient. We can even repeat the operations until the thing gets right. But the mistakes are specifically serious if this is done on the germ tissue. Germ tissue is the only tissue that we have, all the superior organisms with sexual reproduction, in which the gametic cells are generated. So to make operations on a tissue that gametes will derive from, that will go then over to the future generation, that encloses an additional risk, the risk of transferring uncontrolled mistakes to future generations. Once I've said that, the next step would be to give some examples of application in the clinical case, in the medical case, in genomics with therapeutic goals. At present, in 2021, and right now in June, this has been, I looked at this a couple of days ago, in the International Web of Clinical Trials. That's where all these papers are registered at the worldwide level that are being carried out right now on any type of technology with clinical targets. Approximately out of 380,000 researchers worldwide, more than 220 countries, there are 46 papers at present, ongoing papers, that are clinical trials that are using the CRISPR technology with two types of clinical aims, diagnostic or therapeutic. diagnostics simply in order to recognize the presence of the DNA of some microorganism normally, or therapeutic, and this is the aim that we are most interested in, the closest to what the transhumanist wants to reach, that propose to modify the genome. So the therapeutics in this case with health goals are approximately 40-odd papers that are distributed between applications addressed to correcting monogenic diseases due to one single gene that is altered in the patient that gives us then to a pathology. We will see a list soon, or a very interesting application that is as a new method of immunotherapy for cancer. There is one thing that I wanted to say that is important. We speak about a technology that allows to make gene editing, correcting the DNA in details of one gene, one single gene that is involved, because we know it due to the genetic history and many other researches, that it's involved in a disease. What the transhumanists would desire would be maybe to apply it to correct not a specific gene, but they talk more of a feature rather than genes. They talk about features and something that can have a bearing on physical, mental faculties, lengthening the life. So in genetics, this is still starting right now. Genes that are involved in a series of features that are much more complex, because these features are more complex. These are not monogenic systems. It's just, for the time being, still a dream. I say that as a detail for the possibility of implementation of this technology. Among the pathologies that are being tried out in those clinical trials, that are 47 approximately, well, there are some that involve hemophilia A or B, falciform anemia, thalassemia, Huntington's disease, cystic fibrosis, a series of diseases that we know since long that there is one single gene involved, and of whom we know the nucleotidic base sequence, and we also know what zone of the gene is the one that is involved in the pathology. So that's where we have to go in order to correct the pathology. We have to incide on that area. Here we have one case that is known, is relatively recent, how we can correct a gene, a specific gene, involved in the synthesis of a protein, the beta globin. When it is altered, gives rise to several diseases, amongst other falciform anemia or the beta thalassemia or other diseases. There are recent cases where the CRISPR correction has been applied for this type of pathology, the sickle cell anemia. And 1 year after the application, in 2 specific patients, has already been published, they have found levels of efficacy in the editing in the bone marrow and in blood of these patients, and a great improvement in their health. So we have specific applications and for a genetic system that is already known, as doesn't happen if what we want to see other adventures. With respect to the applications for the treatment of cancer, gene editing contributes with a fantastic alternative of immunotherapy. For instance, we have to strengthen the synthesis of antigenic proteins at the surface of the T cells of the immune system, so these antigens are able to recognize antibodies that are at the surface of the tumor cells. So this being a gene editing with CRISPR in a constitutive manner, the T lymphocytes of our immune system can be directly addressed to the tumor cells, attacking them and even overcoming them. This idea of strengthening the T cells by means of CRISPR has also given very positive results. There are even patients who have already been treated with this system, lymphoblastic leukemia, the juvenile Hodgkin's lymphoma, and they also talk now already about the solid tumors as well. Now we are going to what the transhumanists want. to obtain from the research line of CRISPR. Well, what the transhumanists want is interventions in the germline mainly. We have said before that the germline is the one that can go over then to future generations, but you can see a key to modify the human genome with respect to a post-human future in order to induce changes of aesthetic nature or mental nature or of any other type of those that are in the minds of the transhumanists. Ideas that we could call science fiction ones, because many of those features are complex. The system, the complete system of information of the human genome is enormous. We have 21,000 genes and thousands of regions of regulation that we don't know how they work, and thus aspire to modifying according to our will features of this type is really a quite an adventure with unforeseeable consequences. But there they are with their idea of reinforcing the musculature, the use of carbon dioxide even for respiration in an ambient rich in carbon dioxide, like in Mars, for instance. The goal of living without aging, and also systems for repairing genes. But all this is based on a too simplistic understanding of what genes are and exaggerated expectations of the potential of the technologies of editing genes. In spite of the lack of security and the ethical conditionings, there have been several attempts to modify even genes in human embryos. As we have said, in human embryos that still haven't formed the germline, and that thus, when they reach the adult age, they will develop the modifications in such a way that they could even go beyond over to future generations. That's why CRISPR has been received with euphoria by the transhumanistic circles. They want to do interventions in the germline, in gametes, in such a way that they can reach future generations. What are the trials that have been made with embryos? Because this has already started. They are already using trials in embryos. Well, since 2015, in 2015 and 2016, for instance, in university in China, they carried out genetic modifications in embryos obtained with IVF, human embryos, although then they were not implanted.
But the idea was just to initiate trials to see whether it was possible to make that genetic modification. For example, a gene involved in resistance to AIDS was subject to this type of analysis. In 2016, and this was a little bit closer, in the UK, an experiment was approved to carry out genic editing in a human gene with embryos coming from in vitro fertilization in order to better understand the genetic systems that may improve the performance of in vitro fertilization techniques, IVF techniques. And this was a revolution because those studies could have been carried out with animal models. And in 2017, at the University of Oregon in the United States, a study was published in Nature by an important Spanish researcher, Dr. Del Monte, and also somebody else, who also attempted to modify a gene involved in a myocardiopathy so as to correct that gene in embryos. But those embryos which were not implanted either, but the result was very disappointing because they showed mosaicism. Some cells had been edited, others had not. In some embryos, no cells had been edited, and then there were some errors in some other sites of the genome, confirming the risk involved in these technologies. In spite of the lack of safety and the ethical problems, several attempts were made, even in embryos that were implanted. And this is the case of a scandal by this Chinese researcher called Xi Yankui, who performed an embryo experiment in some girls, some human embryos. There were twins, and he implanted them, and then 2 girls were born, with the idea that in that gene editing, these girls would acquire a mutation of the CCR5 gene that would make them immune to AIDS because their father had the disease. Well, as you all know, this was a big, it led to a big outcry, and this scientist was condemned by the scientific and criminal authorities of his country. But these are trials that are being carried out with, I would say too frequently. And this brings me to the end of my presentation, which has to do with the ethical debate. There's a huge ethical debate that, because generally speaking, gene editing through CRISPR poses a series of ethical challenges and a series of questions because of the lack of precision of the technology, because the technology still needs to be perfected. And then in many cases it is not necessary to use embryos, especially when you want to do is how a gene works. You can, of course, use animal models. And, of course, it involves manipulation and the destruction of human embryos, which are, human lives at the first stage of development. And also because there is a risk of transmitting uncontrolled errors to future generations, especially when you follow the germline, or for opening up the way to non-therapeutic purposes, such as, for example, the enhancement of the design of non-health related characters. Gene editing through CRISPR applied to the germline or directly to embryos constitutes today a threat, even for the use of those techniques for therapeutical purposes, because, perhaps in the future this technique may be useful to address different diseases. Given the capacity of this technology to modify genes and genomes in many organisms, in plants, animals, and in man, transhumanists, what transhumanists want is to use it to transform and redesign or direct our own biological evolution, even towards that purported post-human entity with unforeseeable consequences. But there's still a wide bioethical debate, and the main researchers in this field has established a moratorium. The last warning was launched in 2019 after several meetings of experts. In 2019, this was published in Nature. A call was made for a moratorium so that this technology was not applied anymore, especially on human embryos, because this could raise an important question, namely whether the clinical use of CRISPR might be justified for gene editing of the germline and to resolve many of our conditions. Thank you. Thank you very much, Dr. Gobet. You have really provided us with a very good introduction to the subject in a very didactic way. And now we will give the floor to Dr. Miguel Ángel Serra. Miguel Ángel Serra has a degree from the University of Navarra. He's a doctor in biological science from the University of Barcelona. He holds a master's degree in science from the University of Pampa Fabra. Currently, he works for the European Commission as head of the head of the CAS nanoplastics project at the Joint Research Center of Istria in Italy. He's been a manager of research of Pompeu Fabra between 2005 and 2010, and he's been a professor of molecular biology of the International University of Catalonia from 2002 to 2009. He's also a scientific collaborator of the Neuropharmacology Lab at the University of Pompeu Fabra. He's also a member of the Professional School of Biologists in Catalonia and the Spanish Society of Biochemists and Molecular Biology. Professor Serra published scientific articles on biochemistry, toxicology, neurosciences, and international journals of the field. And as an expert in bioethics, he has published 3 books together with a lawyer called Alberto Cotin, encompassing and dealing with the challenges posed by transhumanism. Thank you, Professor Serra, for joining us. Thank you very much for being with us. The title of your talk is Distinction Between Therapy and Enhancement. Thank you. Thank you very much, Maite. First of all, I would like to thank Francisco de Vitoria University for inviting me to participate in this conference. And specifically, I want to thank Elena Postigo, with whom we have collaborated in different activities in the last few years, always in this exciting field of the transhumanist ideology, human enhancement, and the bioethical challenges posed. The subject that Dr. Postigo assigned me has not been changed at all by CRISPR or by anybody else. Distinction between therapy and enhancement, and this is a significant challenge because this is not an easy topic to discuss, but I think that sometimes you need to pick up the gauntlet in terms of these intellectual challenges that may help us advance a little, as the previous speaker mentioned. So, without further ado, I will start my presentation. So, therapy or enhancement? So the first question is, therapy has to do with health and disease, and this is a normal situation in human life, and we've all found ourselves in that situation. But enhancement has to do with honing and deficit. So we're trying to reach a situation which goes beyond what is normal around us. That is, moving towards a kind of ideal, or perhaps we're missing something and we are trying to filling that void or bridging that gap. But what is happening? Well, we have the same techniques and the same technologies that can be used for one and for the other activity. But you may find different models which are invoked by the people using the technologies to do one thing or the other. And this is where the main problem arises. So, if we are sure that enhancement is an intrinsic aspiration in the human nature, and over and above the evolution process, which has a very important role in the natural selection of human beings, in the case of the human being, we have the transmission of knowledge both from the scientific and technical point of view and from the cultural point of view. So you can say to yourself, well, is it legitimate to transform ourselves? Well, Günther Andres said that human beings generate or invent machines with capacities that are superior to those that human beings have, and then they aspire to use those technologies to transform themselves in order to improve themselves, to rise to the challenges, to measure up to the requirements of those systems that they themselves have invented. So that Prometeic envy requires those technologies which Rocco and Bainbridge called converging technologies for improving human performance, NBIC. Nanotechnology, biotechnology, information technology, and cognitive sciences or neurosciences. This is a report that was entrusted to the American National Science Foundation. And these technologies mean, according to Joe Koch, a qualitative jump with respect to the previous technologies. We can't think of a technology like CRISPR or gene editing presented by Professor Gervais as what we had in the past, because this is a really quantum leap, and so the challenge is also a quantum challenge. It's quantitatively different because it means that you can modify or manipulate. I prefer modify than manipulate, right? Because it is more precise. So modify the DNA of germline cells, which in the past was impossible. You couldn't even think of that, but now it has been made reality. It has been perpetrated, right? As some would say, these technologies. Well, here there is comic published in Nature, computational data, then computational data abundance that may be very useful, but it can't be used to do evil. The speed of communication, so anything related to the development of AI and increasing availability of devices to communicate or to interact at a distance with other devices, and therefore you develop the Internet of Things, the world of biology, and then the genome modifying techniques such as CRISPR and others, the 3D printing technologies which have been shown themselves to be so useful a few months ago to design and manufacture respirators. This was done in Barcelona at a car company. Engineers resolved problems efficiently. So instead of manufacturing rear view mirrors, for example, they decided to manufacture respirators for the patients in the ICU. And then robotics with all its applications. So these are the NBIC technologies. These are the tools available to us for therapeutical interventions or for enhancement interventions. So as you can see, they can be used for the two things. So for good things or acceptable things, or for illegitimate purposes or for dangerous things. Whenever people talk about human enhancement and perfecting oneself and transforming oneself, we should look at other places in the world, which are less fortunate than us, and see what the challenges of medicine are. Because when we speak about therapy and enhancement, well, therapy has to do with medicine, right? And what are the challenges of medicine in the third millennium? What are we still finding ourselves? And how do we want to tackle those problems or those situations? So these are the 10 most common causes of death in poor countries and in rich countries, right? On the left, you have the poor countries. On the right, you have the affluent countries, and you can see that the main cause of death in the first countries are infections of the lower respiratory tract, of the lower airways, and many of our fellow citizens have died of this problem as a result of COVID. AIDS or diarrhea, GI tract diseases, because of massive dehydration, for example, infarction, and then ischemic diseases of the heart. This last cause I mentioned is the most important one in rich countries, in affluent countries, and then myocardial infarction, and then different types of cancers, Alzheimer's disease, and COPD. So as you can imagine, the causes of death are very different in poor countries from rich countries. But if you look at the number of people who need health interventions, I'm not talking some health intervention and can't get it, well, water, drinking water is required by 1 billion people in the world who have no access to drinking water. control of hypertension. Here, you go to the chemist and you get your heart rate controlled, but there's many people that can't get that. Also, availability of insecticides. So, the challenge is to transform ourselves using transforming technologies. When there's rehydration kits that are very economical and would save the lives of lots of children, then of course we can aspire to the sky, but we could do other things which would save more lives. As a challenge, I'm saying to improve, our human population globally. This is just to give you some context. And this is linked to what Adela Cortina says. The theory of bioethics the task of bioethics consists in preserving and promoting the life of the present and future generations and of nature, and look after vulnerable beings. And this is a motto that is going to underlie the rest of my presentation. So therapy, improvement, and this is all related to health. What is health? Well, according to the WHO, this is the definition of health. This is in the founding charter. It's a state of complete physical, mental, and social well-being, and not just the absence of diseases or ailments. How many of you would raise your hands to say that you comply with the definition of the WHO? Well, I'm not going to do this. I'm not going to put you to the test. But, well, there's different aspects, very good. But transhumanists, and these are some ideas by the Oxford group that we heard from this morning, to reach that well-being, this physical, social, etc. For this, it is not enough to cure a disease. Enhancers, techno-optimists would consider it legitimate to conduct utilitarian enhancement interventions, which would allow people to live a better life. But they don't define what a better life is. So here, any difference between therapy and enhancement disappears. And a concept is introduced which feeds on technologies, which is the concept of reproductive beneficence, so that the embryos generated by using those techniques may be better, may be benefited, and may be better than they would have been if the intervention had not taken place. And we will see later on how all of this takes shape. But we will see, what are the systems, or what are the definitions, or what are the concepts, the health concepts, apart from the idealized definition of the WHO? Well, the naturalistic school that started in ancient Greece sees the curation of human being as a restoration of health, recovery of lost functions, and it is a non-relativist or relativist approach. But the normative approach, which is more modern, and it says that a healthy person falls within a series of norms, and diseased persons are separated from those norms. So here there's a more active role of medicine. There's an emphasis on form and faculties to avoid or prevent or resist disease. So it's more social and more relativistic. It's a concept of health such as the one used by Gérard Kaplan in 1995. But it can also be seen as a target of medicine. The target of naturalistic medicine is restoring a balance. So, if we are lacking in the case of, well, if we have a mutation in one single area, we may correct that mutation, and then the hemoglobin levels would be normal, and then oxygen would be properly transported. But if we take it beyond that, if we doubled the capacity to transport oxygen, we would not be striking a balance, but we would be going beyond. It would be an enhancement, therapeutic or non-therapeutic, but the idea would be to contribute to add new functions. For example, instead of transporting only oxygen, they could transport some other substance. But in the normative concept of medicine, the goal is that there is no moral difference between a therapeutic intervention or enhancement. The relevant thing is to improve according to each person's aspirations, depending on what each person believes well-being should be for them. And, well, if we carry on, we will see that bioconservationists follow the tenets of the previous approaches, and they distinguish between an acceptable therapy and a therapy which is acceptable, whereas an enhancement which is not acceptable. Whereas the techno-optimists do not distinguish between therapy and enhancement. Both things are acceptable to them. Now, the European Parliament entrusted an expert committee with a report, and this went through an attempt to... The idea was to reconcile both positions. This is a study that was published in 2009. It's called the Human Enhancement Study, and it says that the distinction between therapy and enhancement may lead us to a blind alley, because some people consider it as something that cannot be addressed, but others something that must be addressed. So what they try to do is reconcile those two extremes. So they use different cases in order to find common ground and provide a series of items of advice to then be able to legislate over them. And so what they say is an appropriate definition of enhancement should not include those forms of therapy, and it should take into account that there are limits between medical treatment and enhancement. They are saying that they're very subtle and difficult to define. Some of them are very clear, but others are not that clear at all. And they say that it should not be based upon a strong conceptualization of what normalcy, disability, or health is considered to be. So it's important to be flexible and shy away from that definition of the WHO and take into account the opinion of the people from the vulnerable groups, etc. And then of course, it should exclude practices that consist in the use of external devices, and so the idea is to improve the individual or enhance the individual, but not the species. And this is a table that was published in that report. And this table is interesting because we all agree with column 1. I guess you can see it properly in column 6, but columns 2, 3, 4, and 5 may generate many controversies. For example, some cases that may come to mind. We have two children, right? Tim and Bill. Tim is, his parents are normal stature, but he has cancer, and so he produces less growth hormone, and he is afraid that he will not reach a proper height. So he is given hormone treatment, growth hormone treatment, like Messi, the soccer player, right? Growth hormone treatment. So this is therapy, it's very clear. But there's another child called Bill whose parents are low. They are short, they're not very tall. And so he would normally reach a very limited height. But his parents say, no, we want him to play basketball. We want him to be tall. And so they give him growth hormone. So would the purpose be, that would be a therapy, but the idea would be, the purpose would be enhancement in the case of Bill, right? The purpose of the therapy would be an enhancement. Another case, a disease that reduces muscle mass. If we treat the patient if we give them corticosteroids or anabolics, we would be giving a therapeutic intervention. But if we give it to an athlete to improve his performance in sports, then we would be, well, probably in column 4. This would be enhancement. Another case would be, for example, that we have 2 types of students in primary schools. Let's imagine that you ask parents to give their informed consent, and then one class is given Italium to increase their concentration. This is a drug that is given for hypersensitivity syndrome, but you give it to improve their concentration. And to the other group, you modify the ratio, the teacher-student ratio, so that there's more teachers per student. In one case, this would be a therapy, an intervention, a non-therapeutic intervention, in order to enhance the situation, because children have no condition, and so there's no therapy or no deficit to correct. But in the other group, it would just be an external intervention for enhancement.
We
have the case of Pistorius, but I think that somebody's going to talk about him tomorrow. We've seen already where things are going or heading to. The STOA says one can allow technology treatment that has as a goal an improvement, yes, no. If there is an intentional disfiguration of the human body, if one limits intentionally the amplitude of the human wish, prevents the exercise of human rationality, or prevents the human capacity of choosing freely, or violates the current dignity of people, generate lack of justice, social justice. These are advices that they gave in their report that can be useful for us, but they are not definitive. As far as I know, they haven't been studied in the parliament nor anything else. If we go on with the challenge of bioethics in the 3rd millennium, I also like to focus on the task of a doctor, Pellegrino, who says that medicine is singular for 3 reasons: the vulnerability of the person who is sick. I would say that a human being is also always vulnerable. The nature of medical training, for vocational service for all, and the Hippocrates sentence that teaches them from the ethical point of view. What Pellegrino does is very important in this case for the practical implementation of all these technologies. Adela Cortina told us about the accent on the intentions, and now here we start talking about the very specific intentions with the patient in front of us. If we are patients, he's behind us, and if well, we have seen what this case is, but I would like to point out the definition given by Boström, the moral duty to improving and even reaching immortality even. This is written and published, so I leave it there. Transhumanism, we've seen what it is. I'm just going to focus on the 3 super intelligence, super welfare, super longevity. We will later see what are the specific examples that we can find. I want to stop at what are the contradictions found by the researchers on the transhumanism. We can see the goal of improvement as a moral obligation, but it's not defined. It is just simple, or an ideal, a possibility. Copy the mind in order to duplicate or perpetuate the person. This is not even proven, nor there is no way of saying that you can copy. Well, we see that this is not guaranteeing that you are going to be able to immortalize a given person. At the most, you can keep the information, some information. This doesn't mean that you can copy the conscience. This is something that was asked before lunch. The final goal, the improved man, has been with the traits of the personality are so complex that it's difficult for the technology to reach that improvement in a given trait. And in those technologies, the transhumanists believe blindly, and they say that we have to interfere in the natural evolution. Why? Because the natural evolution is very bad, but with the human intervention and the technologies and this wish for improving, it's going to improve very much. But they forget that there are external causes and factors, both in the techniques and in the environment, and the individual himself. It's legitimate for him to have his own will, the wishes for self-realization. This is his own nature. Aspiring to improve is legitimate, but there is a negation from the transhumanists that is quite clear about human nature. Sometimes they overlook it, as what has been said this morning. It's suspended. This materialistic reduction, but that is renouncing to the bodily nature that we have and to the vulnerability itself of the human being, and then the reduction of the superior dimension in this neo-Gnosticism of the transhumanists to an instrumental rationality. Well, they reduce the mind to the brain and the brain to the neuronal circuits that are going to be set up there. If you copy that pattern of circuits, we don't know how, in the cloud, as Sandberg said, and this is what I am in reality. Well, here the subjectivity becomes the moral norm of what I can do or not do. Apart from that, this is absolutely hypothetical, not proven. I am a scientist, and we have to give time to it. And this is something that is totally biased. They try to sell us something, a motorbike that runs on water. That's complicated. What are the contradictions of the improvement? Well, the scholars have identified some of them. Disadvantage, the resource distribution, the richer ones are going to access before to these improvement technologies than the poor ones. They have an advantage situation that is even greater, and the gap will be greater and greater. Then there is a complicity with damaging idea about the reality. We give an excessive value to a given normality with the social agreement, as happened with cosmetic surgery. We have to reach that ideal of man or woman. So this normality is admired over what is correct. The lack of authenticity, in order to increase their own attraction, the enchanting power, they use the cosmetic psychopharmacology. And then the parental eugenesia that Professor Yue has talked about. And I would like to differentiate the greater acceptability in general about the parental one, but less due to the coercion of the state one. This is Aldous Huxley's Happy World. And the arguments of the slippery slope that would lead to a discrimination, in this case a social one, that would produce an eugenesia towards a dystopic future. I'm going to go quickly over this because we don't have too much time. Possibly, there are a series of bioethical issues that we have always learned and known about that have an important bearing on the type of issue of therapy and improvement. A dual effect. When you can have bad luck, and this goes out of the target. And you improve, some cells improve, and some don't. Principle of responsibility. Hans Jonas knew that we have to be cautious and be humble. Some transhumanists, as John Harris, the humility is no virtue. This is something just to throw away. And I leave it there. Principle of caution that comes from the German school, where they adopted environmental issues. Before damaging, we have to be very cautious, etc. Many people say that it stops the development of science, but it's a principle that has been adopted in the European directives. The problem of the non-identity, what I'm going to do is benefiting an embryo, but maybe can affect the genetic heritage of the future generations, as they don't exist, this is moral, this is not. And then the debate on people with disability that we will see a little later. But in practice, what have we got? Healthcare provider doesn't have the time to read all these treaties. The practical thing are the 4 principles of bioethics: autonomy, beneficial, not good, no harm, and justice. The pharmacochemical improvement. We have drugs, as I've said before, drugs in English, that can be used for what they are indicated, and others. As the class, the one who receives Ritalin to improve the academic improvement, and there are many more other drugs that can be used for something or for a person who is not sick. Those dual applications normally are not regulated. They are off-label. You buy the drug and they sell it to you, and then you think that this dual effect will be produced in you. The bioethical debate is extended more and more socially on if this legal to use them, if it can be done properly, or if there is a trap or cheating that is being created, if it's correct, the off-label use, and where's the border between therapy and improvement in this case. So we have to bear in mind the legal framework, the Spanish Agency of Drugs and Health Products, the training of the healthcare providers, the balance between risks and benefits, and the prevention programs that exist, and then individual and social reflection. The case of intelligences, this is, yes, I'm coming to the end. This is one of the preferred topics for the transhumanists of some, like Fabulesco and Bostrom. We have to make our future generation be smarter. Here is one of the studies of GGWS that says that they have found 538 genes linked with intelligence. A complex character where we have CRISPR. Professor Ube has told her if it's a neurogenic disease, we can start. With what gene shall we start with CRISPR? So you can see it's not that easy. So when we have to be involved in neurogenesis, as the synapses regulation, you know that it's very hard to find where we have to start, how do we have to modify the genes to modify a given nature that is a clear target of the transhumanists. So if we only have to, that 50% of the intelligence is due to inherited features, well, we still see that we are further away than what we can try to modify in order to improve only. So here we have all the concepts of determinism, discrimination, blockade of the information, and then the other thing, the illimited growth. I improved today with this technique, then another one that also gets better. Now I want to improve more, etc. On the other hand, this is what Sparrow calls the enhanced rat race. Well, this generates a situation of unbalance and discrimination that is very clear-cut. I'm going to go over this because my colleague Juan Pablo, I believe, is going to talk about it. It is very interesting. If there are questions, I can answer them as well. And before finishing, I want to show this slide of this person who is Shargaf, who was fundamental for Ricky Watson to be able to formulate the discovery of the DNA and how it is replicated. He's had that sentence just before dying. And he was right. The cell nucleus to touch it is much more dangerous than touching the atomic nucleus, because as soon as you have modified a gene and the gene goes over to the future generations, you lose control of what may happen there. This has been explained already by Professor Dube. Improvement or protective therapy. Well, I would say it's the same. The lack of security, of doubtful, dubious target. We have a red line if this can be transmitted to the offspring. This red line, why it is there? First, because we have to resort to the IVF. Obviously, women are going to be subject to greater pressure and more exploitation because these are different measures. I don't know. More sizes, that Professor Ju explained before, lower efficacy, the post-target effects, off-target instability in the genome, on-target effects with undesired consequences. You hit the target but not properly. The epigenetic events and then the diversity of the genome. If we say we want to reach this ideal for that range, whatever, and for that we have to modify the genes in this manner. Everybody's going to modify it similarly. This in biology is poison because it creates an evolutive vulnerability for the future generations. If we have environmental stressor in the future, find phenomena that are uniform or homogeneous, if they are vulnerable to that, it's the end of them. We have to have variability because the variability allows a few individuals to escape from that stressor because they have in the genetic background the possibility of doing that. Obviously, this is valid for some traits, but not to others. But there it is. And I finish now. In the case of people with functional diversity, we have to ask them, do you want to be modified genetically? Some don't want or some want, as Ukibai's, his family and others want to. But this doesn't mean there is uniformity. We have to ask them. And thus, this difference between therapy and improvement, normal and not normal, we have to be very slowly and cautious because it can be useful to discriminate people who don't have those capabilities and can improve the dichotomy of what is considered normal and non-normal. Interesting bases that you can read in 15 seconds that talk about exactly the disability, if we get better, improve or not. If we improve, we are better than others and the others are worse. So the one that is normal is automatically has a disability. We have to bear that in mind. Shakespeare, who is a person with enormous prestige, talks about control and domination. And then Baylis talks about the collective responsibility for making decisions. All this has to be borne in mind. And to finish the conclusions, the distinction between therapy and improvement can be useful in many cases in the medical setting to make decisions in the clinical practice. But out of this setting, there are a lot of uncertainties of distinction between one thing and the other. We have to have a solid ethical basis. Dr. Murillo says we have to go to the aims inherent to the human being. Transhumanists don't take into account the human nature. But what is important in human being with his own vulnerability, that is very important, and its dignity, not to reach an ectopic society. The transhumanists talk about possibilities and very little of reality. So we have to bear that in mind. And at any rate, the use of the technologies is decided by the society and by each person. And this has to be done with autonomy, with freedom, and responsibility. Thank you very much. Yes, passionating. Thank you, Dr. Serra. And now we will go over to the last speaker of this afternoon and this panel, Dr. Juan Pablo Romero, who is physician, neurologist, PhD on neurosciences, master in biochemistry by the Francisco Vitoria University, and teaches in human anatomy and neuroanatomy for pharmacists, doctors, and biomedicals. And he works at the same time as neurologist in the unit of brain damage of the Beata Maria Hospital, Madrid, head of the research group for rehabilitation of brain damage and disorders of movement in experimental sciences. The field of interest in research are the neurodegenerative disease, movement disorders, and alterations for rehabilitation. The active projects are guided towards the neural rehabilitation, both of brain damage that is acquired as a stroke and Parkinson's disease. With the development of his research, they bring about an effective and non-invasive technique at the hospital level that can be complemented with the conventional therapies, with the goal of improving the welfare of the patient, decrease their requirements in drugs, and also reduce the health cost. More than 45 scientific papers in high-level journals. This activity, no doubt, puts our university in the map of clinical neurology. Dr. Romero, thank you. The title of the intervention of Dr. Romero is Limitations of the Human Brain, Opportunities for Improvement. I will then go over to the podium, but I will start here when I will show a couple of slides. First of all, thank you very much to the Instituto Razón Abierta for inviting me once again to the Congress. It's a challenge always to talk about these issues, and mainly after such important speakers as Dr. Juve and Dr. Serra. Well, in this presentation, probably I'm going to give you more questions than answers, because I can not give you yet conclusions. I have to leave you with questions. And I start by showing this slide with two news. I would like you to think, in what internal reaction do you have when you see those news? The first one is the news that in France they have developed an artificial heart, weighs merely 4 kilos and can be implanted in the thorax. But the difference with the artificial heart that have been previously developed is that this heart allows to be adapted to the needs of the patient. It doesn't pump with a standard rhythm. It can pump the blood with more or less ratio of pumping according to the patient. So this brings it closer to the physiology of the heart. In reality, this device is done for a temporarily transient implantation, but we believe that in the future it may be used. Thank you. I can't give my talk sitting. This way I can show on the slides. This heart is made for a temporary transient implantation, but later on this will be implanted on a permanent basis. Obviously, the discussion could be, as my colleague said, if it's used to alleviate a patient with a heart disease, or if this heart can allow us to live the whole life protected from a heart infarction. And here we have a project that is somewhat old, and I think it has been delayed for the terms that they gave. The Avatar project, in which in 2025 they thought they could have an avatar of the human brain. Totally, the information of neural connections was passed to in silico to an external support. Those two news that cause a different impact in our awareness of what is the improvement, the augmentation, and the technological solutions for some diseases. Why? Because the human brain has a different position with respect to the other organs as to our own identity is concerned. If I'm transplanted a kidney and I don't feel I have changed as a person, but if it interacts with my nervous system, probably I will feel that my person has changed as well. This happens with many types of improvement that can be done in this nervous system. This nervous system that you humans have is not that unique and special. This organ identified as a primordial organ in our system. It's an organ that nevertheless is not exclusive to human beings. And it's an organ that should remind us that we are animals and that we do belong to an animal kingdom in which the brain is repeated in other species with other configurations. And as to the human brain, I would like to call your attention on the fact that we are going to see some structures that are more preserved and are more similar to those of other animals, and other newer ones that human beings could call owned by the human brain. Here you can see the human brain and the rodent brain. We see limbic structures that are the oldest part of the nervous nervous system in the paleo-brain, paleo-cortex, that are giving us the function of allowing emotional regulation. In the function of these parts of the cortex facilitate our survival. Imagine the importance of this function and how primitive the structures that control it. These structures are interrelated with other structures of the body, as is the autonomous nervous system and the endocrine system, that allow our body to be placed in a physiological situation that is different according to the situations of the environment that it is going to face. If we have a threat, the body is activating the system in order to allow our body to adapt and allow our survival. On the other hand, we have in this picture here that I'm putting out the cortex or neocortex. This neocortex, as you can see in the upper part in the rodent, and prosimian, and in the lower part of the human being has different degrees of development. This cortex is the identity imprint of the human being, and where the main evolution is supporting on the cognitive human processes that we call, in a more summarized manner, human intelligence. When we are going to break down what this whole thing about the cortex is all about, I need you to understand things so that then I can explain to you the transhumanist approaches with respect to the use of technology for brain augmentation, if they are real or not. We will see in this cortex that we see in the last column, we have zones in red and blue. The colored red zones correspond to brain cortical zones that are primary. These are zones that mainly are dedicated to one function. Mainly, they are especially sensitive. The primary sensitive zone that is in charge of the sensitivity of the body, the occipital area and the temporal area that corresponds with vision and hearing. The red zones that you see are the motor areas. Let's start making an analogy between the brain and the computer, not because they are similar to a computer, because we humans have invented a computer and try the brain to be adapted to that model. We try to adapt the nature to a model that we can understand. In the computer, we have one way of introducing information. How do we do that? With a keyboard, with a mouse, with a USB. These are ways of afference of information, and the computer delivers to us imprints. This is the way of giving us the efference of the result after the calculation of the process that has taken place in the computer. What happens between afference and inference? We have the magic of computation, the processing of the data. In our brain, likewise, between the afference and efference produced by these parts of the cortex, we see the processing of the information, and it will really be what we can call as conscious mind, intelligent, with many different names. Above all, when they started discovering the function of the brain areas, that is just a simplification according to the last discoveries. Those zones that are in white here, we believe that they will not do function, so much so that you heard that it was conceptualized that only 10% of the brain was used. But this area that we can see in white are zones of association, zones that allow to associate different cortical functions. For a test, for instance, you can close your eyes and you can hear the word mama. The word mama, for many of us, reminds us about sensitive, visual, olfactory, and tactile reminders that are different in each one of us, and they obey to our own insistence. And these associations are there thanks to the cortex of associations. And this cortex is the definitive print of our individuality as human beings. Here we see several animals where, in different colors, the primary areas that I talked about initially and the white areas are areas of association that obviously are much more significant in the human brain than in many other animals. So probably those areas are determining or driving in order to give their capacity to the superior zone. But what is intelligence? This is a very difficult word to synthesize. It's difficult if we define it from a point point of view, a neurological point of view, neuropsychological or biological, or a more operational plane from the point of view of artificial intelligence. I'm going to define it from the point of view of the cognitive neuroscience, where intelligence, we refer to it as cognitive intelligence. I suppose you have heard emotional intelligence as well, but intelligence is really the analogy of the processing of information of the environment and production of a response. As I said, these are simplifications that have been made of the whole cognitive process in order to be able to understand it so that it is understandable. Those cognitive processes, we have two types, the basic and the advanced ones. The existence of the basic processes is necessary for the advanced processes to be evident. The basic evidence probably are shared with the animals, among which we have the perception given by the senses, olfaction, attention is the capacity of processing with preferably a part of information that is perceived or another one, and these are synonymous of the process that take place in the memory. As to the cognitive advanced processes, these are those that we have identified as intelligence, among which we have the most common ones, language, calculation, abstraction, but also we have included other more complex processes that I'm not going to be talking here today. In this conceptualization of the intelligent as a compartmentalized function in different processes, we have here an image that tells us how we conceptualize in neuropsychology the different models of conceptualization. But these are the models used in order to conceptualize the intelligence as different processes that are coordinated and using different parts of the brain in order to produce a response against some stimuli. How does the brain work? Unfortunately, I don't have the response. I think nobody has it for the time being. But let's go into depth about how this processing works. We have a very simple idea. That is the one up there. Information stimulation comes through the sensitive organs, is processed or stored, and then after this there is an answer. But how does this happen? One of the Nobel Prizes in neurosciences due to the discovery of the brain columns. The brain columns in the brain cortex and the occipital zone, the whole brain. The neurons are organized in columns and pillars that share information only with the cells of the column itself. And only in some cells do they share information among them. In those columns, these are complex neurophysiological of inhibition and stimulation that we merely understand. The fact of saying that we still can modify that processing of information is far from being a reality right now. These connections that exist in these columns are evolving. There is an increase in the complexity of this brain because those columns are evolving thanks to the brain plasticity, a concept that many of you have heard about, and obeys to the change not only function but structure of the brain. Certainly now, whilst you are listening to me, you can tell this talk to somebody that has talked about it. Your brain is going to change and your neurotransmitters are going to make so that you can keep in your memory part of this talk. If you hear it several times, the changes will be more stable and will form spines, and you can have structural changes and not only chemical ones. Those changes, the brain plasticity is present in all the stages of life, but in some stages more evident than in others. Here we see that the neuronal density increases with age, but curiously, not only does it increase, they also decrease. We have a synaptic pruning in some of the phases of development in which the synapses that have been formed in childhood, for instance, in the adolescence, many of them are cut. And there is a pruning that takes place and decrease of synaptic conditions because they are not efficient. Inefficient is the criterion that we choose to prune some synapses and not all the others. But we don't know exactly what is the underlying mechanism of this synaptic pruning. These are changing networks and complex networks, not only minute by minute, but also in times of life they change structurally. This analogy has been made more complex every time as we have been developing the world of computation and neuroscience that have been developed in a parallel manner since the posterior phases of the Second World War. They started developing the concept that a computer and a brain were similar. One of the brain characteristics that help to make this analogy is the functioning of a neuron. The neuron, in order to transmit that information, depend on an electrical stimulation or activation. The neurons are depolarized and repolarized, making that this depolarization and repolarization makes an analogy of plus and minus, ones and zeros. And this is analogous to the system of binary codification coding that exists in the computers. So this vision of the neuron as a machine and the construct of the brain as a machine as well have been at the basis for the cybernetics and somehow the artificial intelligence. And many computers with algorithmic systems have based their functioning on some functionings of the neural networks. And the word neural can be found in the scientific publications of engineering. What we can see here is how the concept that we have of the brain has been working. We first have a structural one. We knew that our cortical functions were probably the source of our behavior. And later on, we started discovering the white paths, the axons that connected different areas and allowed to interact among them for a more complex function. Now we are in a phase of the connectome. We are discovering the immense connections that exist between different parts of the brain in order to allow to achieve brain functions. This makes that when the discovery of the genetic code has had a great influence in our way of looking at the problems. We believe that as we have discovered a genetic code, a way of discovering the DNA, we are also going to discover neural code, a way of discovering the way in which the brain structures are connected. The human beings are making analysis of what we already know in order to interpret what is unknown. Probably the brain is one of the fields where this principle is not being too useful. Because, for instance, unlike this hypothesis of active, awake and asleep and ones and zeros, we have then the resting state brain that we use in many of our researches, because that's the brain activity that exists when it's resting. The person is resting. If we follow the principle of 1 and 0, that dual state of the brain as active and inactive, if I'm not moving my arms, my brain and the motor area should be turned off. But when the person is at rest, depending on her baseline condition and the diseases that may affect her, the different zones of the brain are active in a paradoxical manner. So the brain is self-organizing itself without a central control. There are subcortical and cortical structures, and we see that the analogies have reached a limit. And probably it's necessary to create theoretical alternative models that allow us to represent the brain processing as something complex. That probably our own brain is not prepared to understand the way of processing. The cortical neuronal layers communicate in a nonlinear manner. It's chaotic and changing. Not always the same connections give the same result, although we try to generalize. I mean, it's difficult to make independent the voluntary process from the independent ones, involuntary ones. This is important because what I've shown at the beginning, those deep structures that we share with the animals are structures that give an involuntary behavior, instinctive, and the cortex gives a rational, where the brain, the cortex can control and modulate the response to the deep structures up to a certain extent. When the life is at risk, the cortex is annulled and our behavior is still instinctive. I wonder what would happen if there is a fire here or a catastrophe. Probably would not think too much March, what is the best way of going out? We will start running using the activation of our limbic system, our deep system, and we will do exactly the same than if other mammals would be here in front of the fire. We are not different at all. So the decisions we make with respect to this, we have to consider that we do them with a human brain, an animal brain. This brain, with its complexity, has been responsible of all the technological developments that we have been able to do and that have allowed this cognitive leap and this revolution that we have experienced thanks to the development of agriculture, farming, scientific techniques, and recently of the technologies that we are talking about in this Congress. Our cognitive capacity as a species has allowed us to create tools. This capacity of conveying technology through culture is really what has expanded our intelligence. We have to see intelligence as a function of the society and not as something individual, because social intelligence is different that we have as a human group, and the individual intelligence, they are different. But when did we start going beyond the limit? We haven't we done it already, the limit that we have as human beings? Look at those pictures where we see a human being lifting a weight with a lever, and close to that using an exoskeleton. What is the difference between both if these are tools that we have developed and the human being is using in order to be able to work? What happens in the lower part? Isn't it an increase of our memory, an increase of the memory, the fact of writing notes? It's an externalization of the function of the memory, although we don't want to believe it. What is the difference between this and an implant that goes to the hippocampus and allows us to keep a memory in a more efficient manner? Well, it is probable that if we think it in that way, we have already started since long going beyond the natural limits of the human intelligence. Or it's in our nature, the fact of developing these endless tools, so that this allows us for a greater technological progress. The limit of our intelligence, according to what I've said, depends on our tools. The naked brain has many limitations, but we have been for thousands of years doing things that are not natural thanks to our tools. For instance, it is natural for the human being to be able to see a bacteria? Probably not. But when using a microscope, we are able to do that. It means that the use of that microscope really is expanding our cognitive capacity. It's expanding one of the components that we have three of, the perception. The perception is or not part of the general concept of intelligence. Yes, of course. So we are increasing the capacity of perception, and this will bring us to deductive different capacity as well. It's in our nature to look for advantages, comparing with the rest of the others. I've heard that several times in this Congress. Probably our nature is the one that brings us to this limitless curiosity. As you see, slides with many questions and few certainties. How can we increase our individual capacities? And in the rest of society, what is the individual limit? And what are the advantages that we're looking for with these techniques? And one important thing as well is to know that if we not only have reached the personal limit, but also the limit as a species, in order to be able to explain the universe. Why are we eager to improve cognitively? Well, probably by the fact of living in a post-industrial economy where information is power, has a great influence in this wish of improving cognitively that is more and more obvious. The exacerbated meritocracy requires a greater capacity of learning, memory, and emotional stability. The last thing is very important in the recent years. It is seen as an advantage compared with the rest of humans. The acquisition of cognitive skills, investing less effort and time, can be an advantage. This makes us think that what happens if I have to make a career of 6 years to be a physician? Another person in the future can implant information in her brain and can be a doctor within 1 minute. Would we see that a judgment of something that happens through the moral of effort or ethics of rewarding the effort rather than for the adaptation not of having that information from one moment to the other? We look for updated skills and not lose them through aging. This is what leads us to this search. As to the methods of search for cognitive enhancement, we can see that we can analyze them from any point of view. The mode of action, the cognitive component where they act, because there is none acting on all cognitive temporal factors, adverse effects, availability, and social acceptance, mainly. So what are the tools that are available? Tools, many of us use many tools already right now. For instance, biochemistry, nutrition. When we choose one food against the other, we are choosing something that probably improves our cognitive capacity. Drugs, antidepressants, or anxiety relievers can be the fact of having that with the use of an anxiety relieving drug can give us an advantage against another human being that doesn't have access to that medication. Physical implants, electrical stimulation, practically most of the things I hear in talks do talk about this type of means. In my research group, I use magnetic stimulation transcranially, and this can be part of these elements. And behavioral elements, as the discovery that increased sleep improves some cognitive skills, or the mnemotechnic, etc. As to the cognitive help tools, we have tools that we hear about every day, as the wearables, augmented reality interface, brain-computer, the mobile phones, for instance. And I will run over to my time a bit, because one of the things that I have been asked to explain to you that is included in my talk is to talk about Neuralink. Neuralink are devices of brain augmentation that has been the topic of latest discussion in the Transhumanist Forum last year as to the part of the nervous system. Artificial intelligence first, that we have to bear in mind that it tries to fragment the problems in order to analyze them in different fragments and make them more operational. This fragmentation can be atomized and increase exponentially, increase very much the analysis capacity. What are those systems based on that are being developed of interface brain-machine? The interface brain-machine, what it wants to reach is the singularity. This means to erase the limit between the machine and the human. How can we do that? What can be done right now is based on what I'm going to tell you now. This is the motor cortex of the brain, and it has represented in the motor cortex the human being with its different parts. These parts have neurons underneath, and those neurons, while activated, mobilize that part of the body. Since many years, we can do electrocorticographies, put a sheet of electrodes on the cortex that perceive at what point in time there is depolarization of the neurons. So it's analogous to see when this is activated in this part here. What is the difference of development? That electrodes have been developed that are very numerous and very small as to their size, that can be introduced in all the cortex. And we can perceive not only the general movement of the hand, but of the individual muscles of the hand as well, individual muscles of the face, etc. And this has allowed to translate that activation of the cortex into exoskeletal movements and automatic writing and all that, reading your mind. I put it within brackets because in reality they are not reading our mind. What they are doing is replacing the efference point. I told you there was an afference point, the sensitivity, and an efference, that is motricity. What they can do with this is to implant electrodes in the motor part in order to perceive what is the efference. And what happens in half of the sensitivity and motricity is still a mystery. We don't know how they work and where we should implant an electrode in order to know what you think, see how you have reached a given conclusion, or to put an idea in the brain. This can't be yet done. So the detection of the input and output is totally possible thanks to this technology. Obviously, the effect can vary according to the one who uses this, and the acceptability as well, according to the improvements are oriented to improve a human being or bring him beyond the normality. I'm going to show this last slide that I have here, where I ask 3 questions that I believe are quite important. I can't speak about other things because I have been talking for too long. The human being is limited by biology. What other tools of cognitive expansion are we going to develop in the coming years? But I'm certain this will happen. We are prepared now to be able to assume those challenges in the future. Is the human being infinitely perfectible? Or what is the limit that we should aspire to? Where are we going to stop? Well, this is what I wanted to tell you, and any questions, we are eager to answer them. Thank you. Thank you very much, Juan Pablo. Well, we have been running out of time, but it was a pity to lose something of what you were talking to us about. Although coffee was from 5 to 5:30, yes, we have the permission of the organization. We are going to take a few minutes in order to be able to answer questions. Many have already been launched and asked, but if there is somebody in the audience that would like to ask something, or those who are connected online who would like to ask something to any of the speakers, and even be among the participants, maybe there are questions. I believe there is a microphone here in the room, in the audience. Good afternoon. It was a pleasure to listen to all 3. Professor Juve, I would like to ask you, by sheer curiosity and from enormous ignorance from my side, the technique of the CRISPR that you have explained brilliantly well, has it something to do, is it related to the technology of the messenger RNA that we heard so much about with respect to the vaccines? Well, as a technology, it has nothing to do with each other, although there is an analogy as to the fact that the system that CRISPR is based on is the synthesis of an RNA, a molecule RNA synthetic in the case of the CRISPR application. It has a certain analogy. A messenger RNA, as for instance, the one that we all know in the recent vaccines of Moderna and Pfizer, is a sort of molecule that is the product of the expression of a gene. An RNA is always the product of expression of an RNA, and the RNA is something that is derived, it's a copy in a slightly different version of what is the sequence of a gene. And the difference is only due to nucleotidic basis, but at the end there is an end-to-end relation of the molecule that is expressed to the molecule that is this messenger RNA in the case of the expression of a gene, the gene of the antigens of those vaccines. But in the case of CRISPR, for instance, what happens here is that an RNA is also generated. It's not a messenger, but when we talk messenger, the idea is that then this is going to be translated in turn into another type of molecule that is a protein. In the case of the CRISPR system, it's not going to be translated into a protein. It's going to be, this molecule RNA will be used, that is not a messenger, I repeat, to recognize once again a DNA that by basis complementation reaches from outside the bacteria, and we direct towards a specific gene that we want to modify so that they bind, and with that other element, the protein K can break completely and destroys or modifies that sequence that has been recognized by this RNA. So there is a certain relationship, but simply in the mechanism of operation, not that it doesn't have to do with the messenger RNAs that are the product of gene expressions. Here it's a product of synthesis that is not really to generate a protein. It is simply for a recognition of something that comes from outside. I don't know if I have answered. Probably we would need more time, and I don't want to steal space and time to my other colleagues. There is a question from an online participant. No? No? Hello? A question from an online participant that wants to ask Professor Jove on the Chinese, from Professor Jove on the Chinese experiment of 2018. Has there been some follow-up and revealing how those children, those girls are? Thank you very much. Lulu and Nana, yes. I couldn't hear the question, but now I know. What we know is that out of the two girls, one of them received the genetic modification that Cui wanted, the Chinese researcher that did it, but we don't know anything. Well, the other was not modified, but we don't know anything about what other possible side effects the receiving genome may have had. We know that the specific gene that can be controlled, it's a known sequence, the CDR5, I think he said, and we have to study what has been the effect of the operation of CRISPR, and we know that, yes, it has been modified in one of them. And the other ones, I repeat, not. But of the rest of the genome, human genome has around 3,175 million units of nucleotidic bases as 21,000 genes, and great spaces that are not that don't give information, that will never express as a gene. So any alteration in the other regions for the time being maybe is not going to be manifest and never throughout the life, and maybe are evident in their offspring, because they may have altered cells that through the germ tissue go via the gametes to the future offspring. All that can happen. But for the time being, what we know is what we know, and it is very little. In fact, Dr. Cui recognized there was a third baby as well that was not born when his experiment was public, so we don't know anything about it, if that baby was born, modified or not modified. So the Chinese authorities, as could be expected, have covered that with a dark curtain, and the doctor was condemned to 3 years of prison. That we suppose he is in jail, but the results of the experiment have not been published in scientific papers. There is only one video published by the researcher, but it has never been published in any journal, so it has no scientific validity as a research paper. What is not published is not taken into account. Any further questions? The first thing, good afternoon and good congratulations for these very interesting talks. I wanted to make a reflection and question. After listening to the talks this afternoon and this morning, it seems that in the environment we have the idea that after the transhumanism there can be a sort of ignorance as to the human being himself. And I wanted to ask you, what is your opinion on the following? The people who dedicate to research on that, how are we to blame? And why do I say that? Because we are all aware that in all scientific papers and in all projects, always the first part is the marketing, how we sell an idea, always wanting to say that, emphasizing more what we want to sell rather than talking about the scientific idea. And how much responsibility can we have, we researchers, in generating this type of ideas? Well, I can tell you that the researchers, most prestigious research in the field of genetic engineering, have been very cautious as to the possibility of applying the technologies that they have discovered or developed for therapeutic aims and non-therapeutic. And they have said in a coordinated and agreed upon manner their opinion in meetings that have been held at this respect. In 2012, in Napa Valley, they have met, they have had the two summits of the Human Genome Edition, one held in Washington and the other one in Hong Kong. That was where Dr. Cui was invited to explain his experiments. And in all of them, they have shown caution and prudence in applying those technologies precisely in order to not demonize those technologies that they are developing themselves. The researchers are the first people interested in not exaggerate what are the possibilities that exist. And even some that are not researchers in genetic engineering, but that do have a more support role, philosophers or experts in ethics. Sabulesco criticized a given experiment because he realized that that was going much beyond than what he himself was proposing. Probably he did that in order to protect, in order to prevent them from subtypify. But this caution does exist from the researcher's side. And this prudence and caution is absolutely legitimate. I wanted to just add that Karl Popper, who's a science theoretician, said that science involves ethics. And I think that the good researchers are responsible of what they do, and they think about the consequences. A good researcher is always thinking, what consequences will it have, and what will derive from what we are doing. In 1975, in an Asilomar meeting, when the genetic engineering technique started in bacteria, there was a moratorium. The researchers had to stop before going on in something that was very risky. And we are right now in the same situation. The researchers have declared a moratorium to not use the germ cells. I think that here there are external factors, external to the world of science and research, are pulling from these techniques. There is a slogan of the bioethics, not what is technically possible is ethically acceptable. And this slogan arose in that meeting of Asilomar in 1975. That's the slogan of bioethics. Science is ethical, normally. There can be personal ambitions or other connotations or external factors that make that researchers that are not that, like the Chinese one we talked this afternoon, do things that they shouldn't do. I would like to add as well that as to the gender presentation of the public, as to the advances, as to neurosciences, this is what I know. It's not only the researchers to blame, but also the journalists. I believe that debate and the social networks right now lead to a manipulation of the information about the progresses. And for instance, the recent approval of a monoclonal antibody for the treatment of Alzheimer's disease in the States has been totally manipulated and misinterpreted, and the relevance of that approval is minimum clinically, and nevertheless they have discovered the big cure of the disease. This happens in neurosciences. As to this progress, the interpretation of people and what the people believe is happening in reality in science is being changed by the journalistic ethics that work in the massive media of communication that either don't know about science or don't want to communicate it as it is. Well, although it's a passionate issue and there are many things left, many questions in the air, as you will stay in the Congress, I'm certain that any of the three speakers can answer the questions during the coffee. Not today, we have no coffee any longer, but I think it had been worthwhile. Thank you very much to the three of you for having shared with us this afternoon, and thank you to all the participants and those who are here and those who are online for having chosen this panel. Thank you. I just wanted to make an announcement. There is coffee. The free papers will start on time, but this panel in the square, the space in the middle of the campus, there is coffee for the one who wishes. Thank you. Thank you very much.